For the jury
Oh,
to
of course.
arrive, so that was
That's the most important part.
well, you first and then we're live?
Yeah,
Excellent. So I'll just open it again. Thanks a lot.
no worries. Can you go?
Perfect.
On
Thanks a
yours.
lot.
Thank you so much.
Okay, so welcome everybody. We're starting our third session already, and I'm delighted to welcome the speakers in this session, our jury members and the rest of the audience as well. Our first speaker is going to be Cara McKee talking about seahorses, and it's going to be a seahorse themed session. So for now, I'll pass over to you, Cara. Best of luck.
Thank you for the introduction. I'm Cara McKee, and today I'll present my master's thesis involving a comparative analysis of two seahorse species, Hippocampus gutellatus and Hippocampus hippocampus, through visual exploration. To begin, the genus Hippocampus is 46 species of seahorses inhabiting a circumglobal distribution of shallow temperate habitats. They're often linked with seagrass and macroalgae as they're ideal for crypsis and offer great holdfasts. These species are quite important for conservation as they're considered a flagship species. However, their life history traits of low mobility, small home ranges, low fecundity, long parental care, and mate fidelity often increase their vulnerability to threats. Threats such as coastal development, bycatch, illegal, unreported, and unregulated fishing, and climate change has meant that 30% of these species are considered vulnerable on their conservation status, and 41% are data deficient, which means that there's not enough data to determine the population trends or assess their threats. So the seahorse species, Hippocampus gutellatus on the top and Hippocampus hippocampus on the bottom, are native to European subtidal habitats and often characterized as having low dispersal abilities, fragmented populations, and low density. These are two co-occurring species that often partition their habitat, where H. gutellatus prefers more seagrass habitats with higher complexity, and H. hippocampus prefers more uniform habitats with lower complexity. These are two data-deficient species, meaning that with the current information available, we cannot properly determine their conservation status. To decrease the gap in knowledge, it's important to monitor their abundance, their distribution, and their characteristics. But in order to do so, we must first perform morphometric analyses on their populations. So that is the goal of my master's thesis, is to perform morphometric analyses on these two populations of H. gutellatus and H. hippocampus within Portugal using photo identification technique. This will be done first by identifying the individuals, the sex, the pregnancy, and the locations, and then measuring each individual's morphology, and then comparing this morphology between populations, and then comparing patterns found at the population level between species. There are two sites of importance within this study. The first within Teja River estuary. This site is surrounded by subtidal sandy habitat with moderate wave exposure and is characterized with having few rocks and little macroalgae and seagrass cover. From the blue dot and the red cross, there were photographs collected at these areas from 9 different surveys from 2020 to 2023, as well as the inclusion of iSeahorse Photos, which is an online citizen science platform. The inclusion of iSeahorse allowed for the engagement of public participation with science, as well as helped to increase the data for the study. The second site of interest was in the Sato River estuary. The site, which is seen as the green diamond, is also characterized by subtidal sandy habitat, lower wave exposure, and greater seagrass and macroalgae cover. From this area, the photographs were collected from 2 different surveys, again, and also from iSeahorse. So from the locations, the photos were then used for identification of the 2 species using established protocols of I3S photo identification software. This was done by annotating the natural markings on the animals within the images. So for H. gutellatus, this annotation involved ellipsing spots around the face spot pattern. And for H. hippocampus, this involved contouring the core net shape. After the annotation of the images, these software processing and user identification were used to identify if the images were of the same individual or not. So after individual identification, the morphological traits were measured with ImageJ software. These traits include the head length, the trunk length, the tail length, the standard length, which is the sum of those three lengths, and the height. Each measurement was replicated to ensure the error threshold was under 5%, and subsequent body proportions were calculated as the character length divided by its standard length. These calculations were important in the study as the majority of the photos did not include a scale reference, so this included a lot more individuals with the study. Next, after the morphology was determined, the variation in size between location was analyzed, either by two-sample t-test or a Wilcoxon sum test, depending on the assumption, if the data passed the assumptions. For H. gutallatus, this meant analyzing the height within the locations and between the locations, and then looking at the body proportions between the locations to justify any patterns seen. For H. hippocampus, a power analysis revealed that the sample size obtained from the two locations was too small to do any significance testing, so therefore the locations were pooled and height was described through descriptive statistics. Then, to determine if there were any associations between the size variation we see and their habitat, monthly average, minimum, and maximum values from 14 different abiotic variables from both locations were downloaded from EU Copernicus. And these data were used to model any associations with specifically H. gutallatus height. As again, H. hippocampus, those data were pooled. This involved creating a stepwise GLM and principal component analysis. So next, I'll discuss the results and discussion together. From the photo identification, there were 746 available photos of H. gutallatus. Of those photos, 186 individuals were identified, having equal proportions of males and females. There is 28% of the individuals with unknown sex. And so the current literature postulates that there are usually more males than females in populations, and so these results may be consistent with literature. However, further research will be needed to conclude that. For H. hippocampus, 177 photos, there were 44 individuals identified, having more proportion of males than females. This species population structure is less studied. However, a study by Gray et al. In 2022 in Greece and Mediterranean showed that there were more females than males in their population. So again, there is 11% of the individuals with unknown sex. So these results may be consistent with that population or may be different as they are different habitats. Also, just lightly to note, seahorses are weak swimmers, so we would not suspect that they would occur at both sites as they're quite far apart. But the results confirmed that individuals were not resited at either location, so there was no rafting or dispersion, but within the locations there may be some local dispersion. From the photo identification, three individuals specifically could be tracked through 2022 as they showed to be the same individuals photographed throughout that year. These species have small home ranges, so the reoccurrence of these individuals through images shows a characteristic of site fidelity. These individuals are also male, and so their pregnancies could be tracked, where their pregnancies fall within the species' known breeding seasons, as well as the known gestation period. Additionally, there's one H. hippocampus male that was photographed 70 days apart with a scale reference in each photo, so then we could see how much it grew in 70 days. It grew 1.5 centimeters in its standard length, so the head, trunk, and tail. And this is concurrent with literature of its known rearing growth rate of that species. So next, for H. gutallatus, to determine the variation with height between locations, a two-sample t-test, first disregarding sex, was ran and showed that the average height in Tejo is significantly larger than that in Saro here. And then when looking at the sexes to see if there's difference, there is a significant difference for both males and females. Females in the light green, males in the dark green, between Tejo and Saro. So both sexes are significantly larger, and it's also that both sexes are above 18 centimeters, which is the black dash line. So 18 centimeters is the known current maximum height in literature. So that indicates that these individuals of both sexes are largest on record. So then I looked at the body proportions of H. hippocampus to determine if these individuals were proportionally larger. After significance testing, we see that the females' head lengths and tail lengths were significantly different between locations, indicating that males are simply proportionally larger in Tejo, but females are non-proportionally larger in their heads and tails. For H. hippocampus, on average, Tejo is larger than Saro. However, as again, due to the low sample size, the locations are pooled and we cannot perform any significance testing. But it's also important to note that there are 2 male individuals surpassing the 13 centimeters, which is the maximum known height of the species. So similar pattern seen to H. gutillatus. Next, I ran the stepwise GLM to find any associations with H. gutillatus average heights between locations, and found that there was a significant positive association with average dissolved inorganic carbon, minimum pH, and minimum temperature, whereas there was a significant negative association with minimum DIC, average iron, and minimum salinity. Next, PCA was ran on the data to further test the strengths and directions of the differences in height, and found that PC1, which explains 37% of the variation in height, is most influenced by average DIC, minimum DIC, and average iron. PC2 is mostly explained by, explains 29% of the variation and is most influenced by minimum temperature and minimum salinity. And PC3 is most explained by minimum pH and explains 15% of the variation seen in height. So altogether, these 3 PCs and the 6 variables explain 81% of the observed variation. The other 19% is most likely due to some observer bias or some other environmental variation or variable that was not included in the PCA. So to give some environmental context towards these associations, minimum temperature had the positive association with height, and it may be that this range in temperature, the higher range seen in Tejo of the sea surface temperature, could be better suited for growth. Additionally, salinity had the negative association with height. A study by Ora et al. Showed that there was an increase in growth of the species Hippocampus redeye with lower salinity levels. So because there was an energy allocation more towards growth rather than osmoregulation, which is energetically costly. So it may be that these individuals are exhibiting that as well within Tejo. Dissolved inorganic carbon includes carbonate, which is involved in calcification. Seahorses are covered with bony plates, which are a mineral composite of calcium phosphate. So the larger concentration of DIC found within Tejo could possibly mean that there's more dissociated calcium ions for the larger bony plate growth. Additionally, pH plays a large role in the dissociation of ions, specifically carbonate ions. So the lower pH seen within Tejo could justify a larger available calcium ions within Tejo for the larger bony plate growth. And lastly, iron had the negative association with height. Iron does not have a direct effect on the size of seahorses. However, it is used as an indicator for metapollutants, which are growth inhibitors. So possibly the more iron concentration seen in Saro could indicate a more metal pollutants within this area and indicate possibly growth inhibition at this area. However, each of these predictions are quite speculative, and each would need their own research to each location to determine any precise conclusion. Overall, this study provides a first comprehensive analysis of these two species populations within these two areas in the estuaries. And also further proves that photo identification is a valid technique to study the variation at the individual level, the population level, and the species level. And research like this is important for monitoring, which will enable the increase of knowledge of the species and the aid of the development of a conservation status for these data deficient species. I'd like to thank everyone who'd helped me with this research, both professionally and personally, as well as those who contributed to the both beautiful photos. And thank you for your attention.
Thanks a lot, Carol. Well done. A lot of comments on YouTube as well. We'd like to know. So I'll pass over maybe to the jury first for questions, and then if we have time, we'll have questions from the audience.
Okay. First, congratulations for your presentation and
Thank you.
for reaching to this moment. It's just the end of the basket. That's very good, and we're very happy for you. I
Thank you.
have some curiosities, more than I remember. I don't know if it was last year or the year before, but it was also some analysis about the connectivity of the populations using genetic markers.
Of the seahorse populations
Yeah.
within Tejo?
Yeah, in this area, you know something
No,
about
not that I'm
So-
familiar. Yeah. No, not that I'm familiar.
No?
No.
Okay. Another curiosity.
Yes.
The number of individuals of each species that was identified using the pictures
Mhm.
from this citizen science
Yes.
project, in some way give information about the abundances, because one of the key points that you mentioned at the beginning was that it's important to know the abundances of the species, because from that you can consider how threatened is the population.
Yes, indeed. So with my research specifically, I got photos from many different origins, which weren't specifically for this study. So they all kind of detailed different type qualities of images and also different bodies, like what they showed in the photo. So yeah, knowing the abundance is quite important for monitoring. However, the timeline was quite inconsistent, and so I couldn't really justify, oh, I found 186 individuals throughout 3 years, but maybe some are dead, maybe some are like new adults. So the surveys themselves were like more independent, and also from iSeahorse, I only retrieved those within the last 5 years. Those were uploaded to the website, as that's like the maximum age of individuals of these species. So I tried to keep ones that were probably still alive, but it's hard to say for sure. So I couldn't really justify saying this is the abundance in this population at this time.
When you were considering the factors to try to associate the A and the morphometric
Yes.
factors with the factors, was in your mind to try to include some factor related with anthropogenic disturbances at the habitats or something like that? Because I saw that it was lacking there, and maybe it has also an importance.
It definitely does have importance. My main objective of the studies was to just characterize the populations, and so I tried to bring in the abiotic variables. However, there has to be some cutoff on how much I can include in this research. And so I covered just what was available on a layer in EU Copernicus. And so, but it's very important for future research to build off of this and say, okay, maybe these abiotic variables, but also the anthropogenic influencers like coastal development, which is a huge impact, and like both estuaries are have a lot of human activity within each. I do know that this location in my map in Tejo at the blue is quite trash covered. However, for H. guttulatus, it's known that artificial hold-fast use, so what they hold on to, they prefer that over like barren substrate. So within this area, you see a lot of individuals holding on to the trash, which is kind of a interesting point on the anthropogenic influence.
Thank you so
Yeah.
much. I have another question.
Okay, sorry. Yeah.
It's just curiosity. So,
Okay, we can talk after if you would like.
For your presentation and for staying in this.
Thank you. Thank you.
Thank you for your presentation. Very visual and very interesting. I have just small questions. Like Lisa, you already answered one. So the one curiosity was, why did you choose these two species in the genus? Any specific reason or?
For me, no specific reason other than there was this great opportunity that I reached out to my current supervisor last year for studying seahorses, and it was some individuals were used in a different paper by Costa et al. That were looking at the influence of climate change on individuals, like specifically warming. And so he had this great idea for this population, specifically here in Caixo Saudade. And so I reached out to him, and he kind of urged me onto it, and I got very excited from that.
The other question was about the applicability of these results to the future.
Mm-hmm.
You already answered that. So I was wondering if you could keep studying these species in the future, which would be the next steps or to have more information about them?
Definitely.
What would you do?
I would continue to do the I3S photo identification to keep adding on to that data. Also, I would look at different abiotic variables specifically, like their influence, for example, the osmoregulation between, so we see that these were the significant variables that were associated, but within each could be some explanation to why we see this large variation. And so I would collect more of the individuals at each of the locations and possibly go through experiments on each, for example, like looking at the aragonite saturation level at each of the locations to determine if there's like more calcium ions in one area or not. But there's a lot of ideas that I have for that. But just basically gaining more data and being able to actually look at the abundances and have consistent survey dates and consistent photos taken to reduce this knowledge gap.
Okay, thank you.
Yeah. Thank you.
Thank you for the presentation. Also, the aims and scope of your work is very interesting. I think that has a very high potential. I want to have two recommendations. One about the map. The map you show with the Tajo and the Tejostuary,
Yes.
to me has a lot of color and it's tricky to see the point with the cross and
Yes.
the circle, and to me it's very confusing
Yes.
to follow the map.
Yes.
So I try to put it in a clear way, I think. And also, you have to take care about the scientific name, because sometimes the specific name
Is
starts with
capitalized.
capital letters
Yes.
and
Yeah.
capital S without.
Yes.
So
Unfortunate
I think that
mistake.
this is important.
No, thank you.
Also, I have some curiosities. You use the picture from the citizens that took them. How you can ensure that these pictures were taken randomly, or not only of the specimens that are more shallower or more bigger, or?
Definitely. So just in general, the images I got from Tejo, there were 5 in total that I got from the ice innocent seahorse. And through talking with my supervisors as well as the general area that they're shown, I included it in there. But that is also a limitation of this study, showing because seahorses their location is protected. So not for people not to go and illegally fish them. But yes.
And another question in the same way, how did you detect that the different photographs came from the same individual?
Yes,
You have a lot of photographs and then
definitely. Took quite a long time, but through the i3s software for HQ Lattice, that was i3s Spot, and for HQ Hippocampus, that was i3s Contour. So with i3s Spot, for each image, you need to have the same side of the face to make sure that you get the same pattern. But you will ellipse all of the spots up. I did 20 to 40 spots. As if you have more, then it'll become quite specific, as if you change the angle of a head, then it could shift. But basically, the software will take this pattern and then overlay it with another and see the distances between each of the ellipses, and it will create a score on those photos, and the lower the score, the more matching or more those photos match, and then that's where the user identification will come in to confirm if those are the same individuals. And then for i3s Contour, basically the same thing where you have the same reference points on each image. Also, I didn't reference that. You have the same three reference points on each image. And then you will contour starting at the tip to the first trunk ring, and same sort of justification. It's kind of like the individual fingerprint, basically. Yeah.
well, so congratulations again.
Sorry?
Congratulations again.
Oh, thank you, thank you.
Thanks a lot. I think we have time for one question from the audience, if there's none. Anyone have a burning question? No? Well, you've been well quizzed by the jury, so well done, Tara.
Thank you so much.
Oh, this. Oh no. Can you? Yeah. This one. Maybe. Wait. This one.
Yeah. And then this one.
Oh, thank you.
No worries.
Perfect.
Okay, great. So staying on the topic of seahorses, we're going to move on to our second speaker, which is Joanna Oliveira. So good luck with your presentation, and
Thank you. My name is Joanna, and I'll be presenting my thesis, which was investigating stakeholder perceptions and the potential of seahorses as a flagship group in Almada, insights for the conservation of the Tagus estuary. This work was supervised by Rita Sá, Gonçalo Silva, and Miguel Correia. First, I'd like to introduce the estuaries that are very important ecosystems as they provide numerous provisioning, supporting, regulating, and cultural ecosystem services. They are very attractive to humans and often in the center of large metropolitan areas worldwide, which makes them, of course, very exposed to anthropogenic stressors that might decrease their ecological resilience and increase their vulnerability to climate change. However, among other factors, their high biodiversity helps them maintain their overall health. Cyngnathids are one of the many groups known to occur in estuaries. They are present in shallow waters worldwide and frequently associated with seagrass habitats. They are vulnerable to disturbances such as habitat fragmentation and pollution, and previous studies have demonstrated that they are charismatic animals that attract high levels of public support and empathy, which makes them potentially effective flagship species for environmental conservation. They are covered by international conventions, and in Portugal specifically, they are protected by law. In the Tagus estuary, here located in the metropolitan area of Lisbon, there are 5 species of cyngnathids available, including 2 seahorse species, those 2. But little is still known about these populations. However, there are local references such as the logo of the local newspaper or the logo of a local wastewater treatment plant that show that the community might have some relationship with these animals, like a historical relationship. So the objective of my study was to investigate the community's interests and perceptions of the local environment with a special emphasis on seahorses. And this was done in Almada, which is located on the southern part of the Tagus estuary. The objectives included identifying stakeholders, characterizing them, identifying areas of activity and possible conflicts, and assess their perceptions and their local ecological knowledge. This would provide information for the conservation of the local environment and also enlightenment on the role of seahorses as a flagship group for local conservation. So as I mentioned, this work was done in Almada, which has a very privileged seaside and riverside location and is under the direct influence of the central channel of the Tagus estuary that is characterized by generally marine biodiversity. The temperature here is mild for much of the year, which favors touristic and recreational activities, but other activities encompass industries, small-scale fisheries, and also fluvial transport. My survey points were these 5 points that were chosen because they were easily accessible and also representative of the study area because they encompass a lot of activities. Okay, my study was based on semi-structured interviews. And to be included in the study, participants had to frequent the area or be part of any organization that had any kind of influence with the Tagus estuary. To start, an initial list of stakeholders was compiled, and then in-person and online interviews started. The questionnaires were divided in 5 sections. The first section included questions about participants' socio-demographic data. Then the second section was specific for fishermen. The third section asked for participants' perceptions on the local environment and the seahorses. The diagnosis section asked for questions that asked participants to identify strengths, weaknesses, opportunities, and threats to local environmental conservation. And in the 5th section, participants were asked to name other participants that should be included in the study to fit a snowball sampling methodology. All map-based questions were based on that map, in which participants could point out the numbers in which they carry out their activities, or in which they have spotted seahorses. All map layers were generated in QGIS. The quantitative analysis was done in SPSS for a multivariate GLM, and the rest, and most of it was done in RStudio based on Fisher's exact test, carried out, followed by pairwise comparisons in case of significant results. Some data was only analyzed qualitatively. So this resulted in 100 participants being interviewed from six different sectors of activity. And here I'd like to clarify that the fisheries sector included only professional fishermen, and the recreational activities sector was mainly represented by recreational fishermen. They have both been fishing in the area for decades. Fisheries are a source of income for most professional fishermen, but also for some recreational fishermen, and most recreational fishermen come from families with fishing traditions. Here, we can see the percentage of fishermen that fish with each gear type, with cane and handline being the most popular method overall, and nets also being widely used by professional fishermen. I'd like to draw your attention to these 2 methods. Hand catch while diving is completely forbidden in a Tagus estuary, and therefore used by both professionals and recreational fishermen. And dredges are not illegal, but they are limited to 30 licenses, so people generally use them illegally. As for the species, there is some overlap. However, Professional fishermen tend to focus on high market value species such as octopus and meager, and looking at the locations in which they have reported fishing, showing darker colors showing more fishermen reported fishing in each location. We can see that professional fishermen fish throughout the study area, especially on the western side and in front of the fishing port, like that dark red. And here, recreational fishermen tend to fish from the coast in easily accessible places where there are pontoons available. Fisheries have a very important role in supporting local livelihoods, but the fact that recreational fishermen also, some of them also rely on fisheries as their source of income, raises concerns about the sustainability and the suitability, of regulation and enforcement measures. And this has been leading to the establishment of illegal activities, which is a major problem in the Tagus estuary. As I mentioned, dredges and hand catch while diving are mostly used illegally, and they are mostly used to catch Manila clam, which is an invasive species, but the problem is that the whole system is illegal, and the harvesting of this species competes with sustainable practices. And also participants mentioned the emergence of illegal activity that employs chemical explosives to catch meager. This, of course, harms the species, but also the surrounding habitats, especially bottom habitats such as seagrasses that are very important to seahorses. As for the perceptions of participants about the environment, we found out that scenic beauty is considered to be the preferred overall attribute selected by participants, but sector did play a significant effect on the selection of attributes by participants, with fish resources being significantly more selected by professional fishermen than all the other sectors. Also, when asked for three words that come to their mind when they think about the study area, participants mentioned landscape, well-being, which are, again, with scenic beauty related to cultural and aesthetic ecosystem services, as opposed to professional fishermen that mention more fish resources and income that are more related to provisioning services. And this, of course, highlights the need to include both in environmental conservation plans in the area. Also, there were identified some conflicting perspectives about, for example, the wastewater treatment plants were considered to be a strength for environmental conservation, but also a weakness, as mostly professional fishermen identified, say that they cause a depletion on fish stocks, but this is, we know it's not correct because recent studies have been shown enhanced biotic communities around these plants. Also, pollution and community awareness levels are considered both as strengths and weaknesses to environmental conservation. And this is mainly because the Tagus estuary is much better than it was a few decades ago. But there are still a lot of problems with pollution. The participants reporting a lot of little debris in the bottom, and also ongoing issues with illegal sewage discharges. Finally, getting to seahorses. Here in this graph, we can see the percentage of stakeholders per sector that have or have not seen seahorses in the study area. So basically, we can see that most participants were aware of the presence of the seahorses in the area, but professional fishermen were significantly more likely to have seen them there. This is mainly because seahorses, as we all know, have a cryptic ability, so they tend to blend with the surroundings, and therefore their sightings were mainly related to bycatch incidents. The gear mostly related to these bycatch incidents were nets, especially gill nets and dredges. And this has been identified as a very representative or important threat to small fish species such as seahorses. Participants didn't have a clear perception of the abundance of the population over time, but they did report sightings across the study area, with most of the sightings with bigger circles occurring on the western part and overlapping with high fishing density locations, which makes sense because bycatch. Also, divers frequently mentioned seeing seahorses clinging onto rubbish, which clearly shows their ability to coexist with anthropogenically influenced habitats. Finally, again, we ask the same question: name 3 words that come to your mind when you think about seahorses. And despite some differences between groups again, there were some words such as charisma, words related to charisma, nature, and conservation that were very frequent, and we didn't identify any conflicts between groups, which goes in accordance with what I said in the beginning, that basically says that these animals are seen as very charismatic species and attract a high level of public support. Also, a lot of participants mentioned stories involving seahorses, highlighting their they have like a kind of fantastic perception among participants, and a lot of participants have reported owning a dried seahorse at home. Which of course has conservation problems because they don't target them, but when they come in the nets or as bycatch, they keep them at home as like an amulet for good luck. And yes, there was also a report of the trade of these animals for the traditional Chinese medicine market. But it was an isolated report, and we couldn't really conclude that it happens in the Tagus estuary. But we know that it does happen in southern areas in Portugal, in Rio Formosa and Faro. So we need to not discard that hypothesis. So I think throughout this presentation, the takeaway message is that preserving the Tagus estuary yields benefits for the natural environment, but also for humans' well-being. And here, I think we showed that seahorses might be a potentially effective flagship species for local environmental conservation, as the participants showed consensual perceptions, no conflicts, they are present throughout the study area. But to ensure their long-term sustainability, it is necessary to implement some protection measures, research and monitor about their population dynamics, but because we still know very little, mitigate threats, eradicate illegal activities because we know that they're being emergent in the Tagus estuary and very harmful for seahorses. And also, the most important thing I'd say is like to collaborate with local communities, especially fishermen that showed the highest interest and influence in the local environment, as well as the highest local ecological knowledge, to know more about this population and incentivize them to care about them. Finally, I would like to thank my 3 amazing supervisors and everyone that helped me throughout this project, especially all the participants and the fishermen, because they were really the foundation of this work. And thank you.
Thanks a lot, Joanna. Well done. Nice presentation. So I'll pass to the jury for their questions again.
First of all, thank you for your presentation. It was very very interesting. I think that the aims you have and also the scope of your study is very important.
Thank you.
I will have a recommendation, a general recommendation. I think that perhaps it should be good to reduce the text on the slide as much as possible because it's easy to follow if you put more picture or diagrams.
Yes,
A
yes.
bit, okay, I have a question. You mentioned the Manila plant,
Yes.
the Japanese plant. Do you think that this species may affect the dynamics or the population of the seahorse?
I don't know. I think it's a little bit maybe far-fetched to say yes or no because there are no studies about this, but we do know, like what we do know is that the fact this is a very widespread illegal activity throughout the estuary, and they're using like hand catch while diving is not the problem, I think, because it's more selective, but dredges are really uh, destroying the bottom, and this of course has impacts on the seagrass especially and other bottom habitats that are important to seahorses. But I cannot say that the presence of the species itself, because I have no information about it to be honest, but it does have influence on other species of clams that are now mostly completely extinguished in the estuary. So
Yes, so
maybe.
maybe indirectly they.
Yes.
Also, I think that this is very important to include local communities and also practitioners in your study. That is a very good point to show. And do you think that if your target species was not seahorses, that is as you call a flagship or a sex species like sea stars and another one, if you study a snail or a durian, a worm, it has the same impact.
Seahorses, like in this specific community, they hold a special significance because, I mean, a lot of, especially fishermen, attribute them like good luck and prosperity. Other people are like, wait, are they fish? And you know, like they don't really know what they are, and they're kind of a fantastic, animal. Um, so I think this species specifically is a very good species for the Tagus estuary local conservation. And I know I don't know about any work that is like assessing perceptions about dolphins, for example, but dolphins are coming very frequently to the Tagus estuary. But some fishermen mentioned to me like, yeah, but they hunt the same species as we fish. And with these animals, they don't have that problem because they don't target the same species. So yes, I don't know about snails. I'd say my answer would be no. Like it's not, these are more like attractive, but
Okay, thank you and congratulations.
thank you.
Thank you for your presentation. I have just one comment. You focus on the fisheries and fishermen, but I also think that it would be interesting to ask about this, about perception in the society. And well, you did,
Yes.
but like more with more focus on the civil society because this is a part of the population that is living there, and also regarding the future, if some monitoring or some procedures are going to be taken, this is a very important part of the
Yes.
state that will also consider this modification. So maybe to take into account not only fisheries and fishermen, but also the other part of the population living there would be also interesting to compare maybe different groups.
Yes.
Young people, old people, and people with studies, or maybe that would also be interesting and you could also see differences.
Yes. I did interview people from all these sectors. Of course, I didn't have time to present all my results because as I did a questionnaire, I had results for each question. And so I tried to focus on fishermen, and But it's true that in my study, like when I present this graph, for example, and I say that most participants know that they are present, this is not representative, of course, of the study area of Almada municipality itself, because I targeted stakeholders that had influence and were very related to the estuary. So of course this is a bit like maybe biased towards people that really work on the estuary and not like the whole community. But yes, that would be very interesting. But I also had like one of the limitations of my study was that not everyone was as willing to answer as fishermen, and fishermen was easier to just go there and ask them. And considering the time I had, this is what I got. But yeah, that would be very interesting, and I'm sure the results would. I mean, I'm not sure because you can never be sure, but like I think the results would probably change a little bit if I included more people from the civil society. Yeah.
Thank you for your presentation. I mean, all about seahorses is beautiful. It's crazy. This is not a tricky question, okay? But what do you think about your sample size? I mean, it's good, it's low.
Okay.
Tell us something about the history about that phrase, the time. I mean,
Yeah.
it's just to listen your opinion.
No,
Comparing
yeah.
with other studies, or I mean,
Yes.
just to evaluate how
Other
much.
studies, usually these surveys are not done in 6 months, coming back to the time, not coming back to time, they're done like in several years. And I think this was a good sample size taking into account the time, because like some of the interviews took like an hour, others took like 15 minutes because they were semi-structured interviews. So I asked questions and people would and people would just talk as much as they wanted. But of course, I'd like to have first more of a balanced. They're quite balanced, but like still more people from like the sectors from which I have less people, and also a big sample size from each sector, because of course the like statistics would be more, accurate, I guess. But this is never perfect, I think. Yeah.
I would like to ask you about other studies in Portugal. Because what you're proposing is like, okay, this can be a way. I mean, to use the seahorses as a flat species to try to protect the estuary. Are there other successful histories in other estuaries using one flat species and then starting from that point to organize a campaign to protect or to establish a new field or protection in this area?
I think like this is a topic that is emerging worldwide. So there's a lot of like not a lot, but some literature about the potential of seahorses as flagship species for specifically estuarine conservation. So yes, of course, it won't solve every problem of estuaries, but I think it's a start, because if people know that there's something that they care about in there, they would want to protect. And in Rio Formosa in Faro, like south of Portugal, yeah, it's like the most studied seahorse population in Portugal, and they do have some campaigns, and they have citizen science, as Kara mentioned, and yeah, so there's some projects going on, but I mean, I think yes, they have potential. Yes, there have been seen some positive results, but saying that they will solve like from bottom to top every problem, maybe no. But yes, we have to start from something, I guess. So yeah.
I was very curious about. You say, okay, I mean, when you put the figure about how many people have really seen seahorses,
Mm-hmm.
I mean, there are a lot of people that never seen seahorses, but I mean, it's kind of contradiction, no? They have never seen the seahorses, but at the same time, many people know by history, no, that they
Yes.
were there.
Because as I mentioned, like I surveyed here, and this is probably the biggest fishing community in Almada, and a lot of the people that are living here are like, they have cousins that are fishermen or parents that were fishermen, and fishermen really have this beliefs of like seahorses being for luck and like bringing them home as an amulet. So people do know that they were present even though they have never seen them. But if you go like maybe here, the fishing community is smaller and people are not as aware. For example, fishermen that have long history, like family tradition in fishing, they are mostly here and they remember, they have stories from their grandparents, and I wish I could have shown them all, but I didn't have time. So yes, I guess even if they haven't seen them, they have heard stories and
The last question. No, I have many questions, but it's just the last question is, what do you think that you will get if just I study? For example, when we are working on sustainable fisheries, in all the surveys we always include one question that is, are you in the will to pay more? For example, if this fish is coming from a sustainable fishery, I don't see your survey, but I don't know if there is some question that is related with that in the sense that, okay, to the fishermen, okay, are you in the will to limit your activity, or, I mean, the influence that can have to protect the estuary through, from the seahorses protection using seahorses as the flag species. And I don't know if you in your survey include something like that, because this open windows that are very interesting for the scientific and for the research community to work on. I mean, to try to modify that way of thinking. No? Because sometimes protecting something is that you must change a little bit your activity, or you must change your habits. I don't know if you include something like that in your survey.
no, I didn't. Because I had like at first I had no idea about what to expect, because I was expecting that people didn't know that they were there, and I was very surprised that they were there. So I didn't have any that they know that they knew, but so I didn't have any questions on that. But from what I got from fishermen specifically, because they are the ones that have most contact, they say that these practices of like, for example, drying seahorses were like, they're not as common now as they were before, and some of them even told me like, yeah, like I want to see them in the water, not in my shelf, like. So, and I even went to some fishermen houses, and they were like, I have this, like, I know it's not right, but it was a lot of years ago, and I don't do it anymore, and now we return them to the sea. So it's like, I think like they're shifting their mindset, because also this is attracting a lot of like, maybe like attention to this area, also because there was a pontoon collapse here like last year, and it was very mediatic, and people were very like interested on the seahorses there as well. So now they want to protect it because they think it's a good thing. Most people, as I showed from my perceptions. But regarding the fishing activity itself, I'm not sure if they would be willing to change their gear, but it's also because they are under so much other stressors and pressure, like all the illegal activities that compete with their own activities, and so it's not easy to answer that, I guess. But that could be.
Thank you so much.
You're welcome.
Thanks for answering the call. It's very nice as well to see on YouTube plenty of support coming in from your research group, so well done. So that's it for this session. Thanks to our two speakers. We get to go for coffee a little bit early. So well done again to Cara and Joanna. And so the next session starts at 5 p.m. Yep. I'm free now. Sharif, I'm good now, anytime. I'm free.
everyone. My name is Frida, and I'm happy to present my thesis project about the characterization of the coral reef communities in Nasisacatia, Madagascar, by using the Reef Check Tropical Protocol and structure from ocean photogrammetry approach. So first of all, I would like to introduce the importance of these ecosystems, which support 35% of the global biodiversity, but also for human well-being by providing food sources, coastal protection, and also income activities such as in the tourism. And even though these ecosystems are well known globally, there are a lot of threats and that can cause the declining of this ecosystem and associated fauna as well. So sclenorctinian corals are colonial organisms that have a special relationship with a microalgae called Zooxanthella, and sclenorctinian corals have the capacity to build complex habitats that can host diverse species and amount of organisms, and has been well described that the loss in the of these habitats in the complexity and their structure have direct impact on the diversity and the abundance of these species that lives there. So it's very important to study them. Particularly for Madagascar, in the northwest region, it has been described a high diversity, one of the most diversity regions in the western Indian Ocean. And there has been some studies that describe an overall good status of these ecosystems. However, there are not monitoring projects that can help us to study them in long term to see the changes. And particularly for Madagascar, there are local threats such as poor waste management, also increasing tourism and overfishing. So considering the importance of these ecosystems as well as the local traits that exist in this region, the aim of this study was to characterize the coral communities and associated fauna in 5 different coral reefs located in Nasisacatia. We examined the associated indicator species of fish and invertebrates with the Reef Check Tropical Protocol, and we describe the coverage and structural complexity by using photogrammetry. So our study area is in the northwest of Madagascar and the archipelago of Nasibe, and this tiny island called Nasisacatia, where we study 5 coral reefs between September and October 2022. So what we did on these sites was study the associated fauna by scuba diving, where we deploy belt transects with 3 replicates per site, where we identify health indicator species for invertebrates and fish that help us to identify some human pressures such as overfishing or harvesting of invertebrates. We study the substrate composition by using photogrammetry, and we use exactly the same study area as the one mentioned before. And we take pictures in this area by ensuring the overlapping of consecutive images. And afterwards, these photo samples were analyzed with AGISAF MetaShake to reconstruct 3D models and 2D orthomosaics that we use afterwards. For the substrate composition, we characterize it by using the 2D models, 2D orthomosaics, and we use QGIS but to select random quadrants in this orthomosaic, where we digitalize polygons and we calculate the area. We identify the substrate type according to the Reef Check Tropical Protocol, and also the hard corals growth forms according to Veron 2000. And we also use the digital elevation model to assess the structural complexity, where we calculate 2 habitat metrics, the structural complexity, and the vector rudeness measure at 3 different resolutions. So the experimental design of this project aims to test differences between the sites and by focusing on the abundance of fish and invertebrates, coverage of benthic communities, as well as cultural growth types and structural complexity. So we use the software of Primer, and we use analysis of permanova and a posteriori pairwise comparison when it was needed. And we also conduct a simple analysis to know the species contribution to this dissimilarity or similarity. And we also did multidimensional scaling analysis and gap analysis to visualize the dataset. Particularly for the structural complexity, we use the software of PAST, and we conduct non-parametric tests. So moving on to the results of this research for the associated fauna and the fish assemblages, we found 17 indicator species listed in the RIP-check tropical protocol. The most abundant ones were Rudhanus and Catodontidae, in contrast to other two families, Amulidae and Serranidae. The analysis of permanova showed an homogeneous abundance among the sites, and this can be explained by the close location between the sites, which has been previously described like that the movement pattern of a fish can be very short or very long, depending on a lot of factors such as species, size, density, and so on. And if we look into the most dominant families that we found, they have particularly long distances for the larval dispersal. So this can give us an idea about the ecological connectivity and exchange of individuals that are in these reefs that we study. So according to the RIP-check tropical protocol, the high abundance of the family Catodontidae can indicate that there is an absence of overfishing for this particular family, and the low abundance of other indicator species can suggest overfishing for them. However, even though in Madagascar has been very well reported and poor management of fisheries, there is unavailable information to compare and to define the current status of the population of fish. For invertebrates, we found 3 indicator species, being the most dominant the sea urchin diadema and the sea urchin, the sea cucumber Oroturia edulis, and followed by the mollusk Tridacna. The analysis of permanova show heterogeneity between the sites, and according to our simple analysis, the abundance of the diadema was the one that contributes mostly to the dissimilarity between the sites, and an overall over-harvesting in the region for invertebrates has been reported in Madagascar since time ago, and particularly for sea cucumbers, the stocks of these organisms have been considered as depleted and near to the collapse, and this is due to the existence of Chinese fisheries, but also foreigner fleets in the Northwest region that affect these populations, and this kind of support what we found in our study with the low abundance of sea cucumbers and other couple of 2 indicator species. For the mollusk Tridacna, we found a couple of studies that describe their density. These studies were conducted in different regions to where we conduct this study, and the values, when we transfer them to what the area that the units that they use, it kind of look like higher than what they found. However, we cannot compare it according to the area, because this was conducted in the southwest, so the conditions are super different. And for the sea urchin diadema, we found very similar study that was in the Nosisakati Island too, and the values were very similar to what we found. And these authors compared these values with the other heavily fished areas in Kenya, defining them as low values for the region. About the substrate composition, we found a dominance of hard coral coverage and rock among the sites, and contrast to other type of substrate such as nutrient indicator algae and other organisms too, but this could be due to the limitation of the quality of the pictures to identify tiny organisms, which is very difficult for algae and tunicates, for instance. And two previous reports describe like similar percentage to what we found, defining them as good conditions in the region. The simple analysis describes that the type of substrate that mostly contribute to the dissimilarity between the sites is hard coral, rock, and sand. And this can give us an idea about how, even though on a small scale, the conditions that contributes to the coverage or the substrate in these reefs can be due to different factors. For instance, there was one site that present like a particular topography, which was very flat. It was like a plateau, and the other ones are like pinnacles. And could be interesting to think about other factors that contribute to the coral coverage, because this site has the lowest coral coverage value or percentage, and it also has the higher rubble percentage. So this has been used as an indicator damage, for instance, for tourism activities with anchoring boats. So it could be interesting to look into this factor. Regarding the growth forms, the most dominant were columnar and massive, compared with two other different type of growth types. And the similarities between sites were due to these four type of forms: columnar, massive, branching, and free living. And regarding the complexity assessment, we observe kind of different values between the sites. For instance, these sites over here, it has like higher values, and this is due to the high coverage of branching and arborescent forms, which are more complex. And in these sites where the values were lower, it presents other simple forms or other type of substrate that doesn't contribute to in the same way that we will expect for branching corals, for instance. However, the non-parametric test that we use, it wasn't significant. And this was kind of similar to the roughness measure that we use for the 3 different resolutions levels. And we saw similar pattern between our sites. And this could be due to the diversity of growth forms, but also the substrate type that are in the corals. For instance, for the sites that have branching and arborescent corals, can have like certain complexity, but for the other sites that have only or mostly columnar and massive, they also have certain complexity that kind of have the same function. And for instance, the site that has like low coral coverage has high rock coral coverage. So the rock was also contributed to the architecture of the ecosystem. And there are some authors that suggest to include other indices to complete this study. So to summarize, this study characterized the coral communities in associated fauna for the first time, and a baseline is defined with this. For the fish assemblages, the overfishing for some indicator species is suggested. For invertebrates, there is also overfishing for some invertebrates, but further studies are needed to define the current status for the mollusk Tridacna and Diadema. For the Bantu communities, we found these three different dominant substrate types, but an overall good status based on previous study can be defined. There are influence of other factors that are needed to study. The structural complexity is due to the diversity of growth forms and substrate types that contributes to the ecosystem architecture. But of course, there are further indices to explore. And I would like to thank to all the people that contribute to this research from the field to the processing of these data, and that without them this couldn't be possible. So thank you so much, and
Thank you very much. Within 20 seconds of the time, so that's considered in the margin of error. Really, thank you so much. Yes, our host, would you like to start?
Yes, I can start because I'm no specialist on this, and so I have to use this. I have only, well, first is to congratulate you for your presentation and for being here in this last step of the master. That's very good. We are happy for all of you now presenting your results of all this time here. You are talking about, maybe I got lost, but what is the reason behind selecting these five locations in Madagascar? I mean, you said, okay, I want to characterize these five locations. Why? I mean, why these five locations? Because you have a previous hypothesis about differences in these locations, about differences in anthropogenic influence. What is the reason behind selecting these five locations?
well, these locations were chosen mostly because it was where we could have the facility to do the research, but there is actually not any previous study in this island. So that was the intention, that to do this project to explore these ecosystems, and especially due to the importance of food security for the people that lives in this region. So fisheries is one of the most important threats for the country, and just few studies have been conducted in the region. So we just had the opportunity to conduct the study, and the sites particularly when chosen because our collaboration with the people that we were working with, the distance, we have other sites around, there are a lot of coral reefs, but they were the most feasible in terms of organization for the project. So we didn't choose them for other particular reason.
I have another short question. I guess about the reef check protocol, the tropical reef check protocol. Because I mean, in other experiences, when you try to apply something that is on the consensus, but sometimes it's not. I mean, when you try to apply the protocol, sometimes it's not completely adequate to your situation. Was that the case in your case? I mean, after this work, you think that there are things that for your specific area maybe are not completely, or it is something that, okay, this is the consensus, this is what we don't want. Maybe other species can be more indicative for this area, or what is your opinion about that? Or after this work, what is your thinking about this?
That's actually a good question. Because this protocol that we use, it's for the tropical regions, it's like used in general. Nowadays there are the same protocols but focus on very local conditions, for example Cambodia or I don't know, Philippines. So they are adapted to the local conditions. So when we use this, we use the general one for tropical regions, which means that the species listed on this protocol were defined by the scientist, and because they were, for example, species that are globally overfished in these regions. So I do think that when we use this method, we realize that the protocol should be adapted to the local needs of this region, because even though in other areas of the tropics there are certain species fished, doesn't mean that in this region is also fished. So that was something that we definitely observed by using this protocol, and we think that should be definitely studied in the region according to the local needs and local context.
Thank you so much.
All right, thank you. Very
Yes, okay, yes, thank you. Nice presentation. Just following up on what Iyaz was saying about indicator species fish, for example. This protocol, you said, is for the whole, what, Indo-Pacific region, I suppose, or for all tropical areas? Or?
See, Indo-Pacific, like it says all tropical areas, and then it includes list of species that classify per regions, for example.
By region, okay.
Per
Because I was
regions.
thinking, why would you expect a particular species to be an indicator for this area when maybe it you know,
Mm.
you understand what is basically what yeah,
Mhm. Like, I mean that, like, this one, like, it's for the Indo-Pacific, no, the Indian Ocean in general. But I do think that when the species were defined, that is a very also old protocol. They could also have been now other species that are being fished in this region, because maybe there are not the stocks that were defined in the past, maybe they are not existent anymore, or maybe they are very low and now there are other species being fished. So this is why I mean that would be useful to take a look to the local conditions.
I have a question about the methodology used in the field. These underwater search or sensors. How do they deal with the cryptic species? This is very complex. There must be a lot of species that are hiding and that are not even easy to account for. So
You mean for fish?
yes, fish.
Well, this protocol is based on citizen science approach. So all the species included in this protocol are species that are easily to identify, and they are not include cryptic species. So we didn't have any problems to monitor them because they were very easily. We have to learn it because we were not familiar with the region. So we have to study to learn the species, but all of them were easily to identify.
And in one of the very first slides, you meant, well, it's written there, I don't know if you mentioned it, size spectra. You didn't look at size spectra. Yeah.
Let's see. This
Yeah, then that slide, you have size spectra. Can you just explain what is going on there?
Well, here according to this figure, the size spectra, I think it means like the size of the fish associated to the coral reefs and the complexity. And how does it change when the habitat is lost. And I use this figure most to show what happened with the complexity or the habitat decrease, more than use it for the size spectral or something regarding with the fish size.
Okay, I'll come back if there's more time.
Thank you. Nico?
Yeah, well, congratulations for the work and for the presentation. It was really nice. It flowed very well. I understood pretty much everything. And about some formal aspects, I would say that some of the layers of the figures are too tiny, so we couldn't see it. And that could be a problem because, for example, can you go to the MDS for multivariate analysis for the invertebrate community?
Sí.
Yeah. So there, to the top right corner, you have the stress level of the MDS, and that's kind of important to report for this kind of analysis. So I cannot, it's difficult to see it. So some layers of the figures are not important, you can skip them, but some other details are important and you must show them properly. And about this MDS, how many invertebrate species do you have for this analysis? Do you know more or less?
How many invertebrate species?
Yeah.
Species were only 3.
3?
Yeah, like, because we follow this protocol and there were just few species of invertebrates, we include only what we found. Yeah, so this was the 3 species that we found. And it was the abundance of these 3 species.
3 is a little bit too low for multivariate analysis. Usually 10 or more species are required to get a proper pattern in this multivariate analysis. One last question about the structure of the coral reefs. You mentioned that you have some places that it was more branchy. You have more. You have species with more structural complexity, and others were more simple. No.
Yeah.
How do you explain that?
Well, for instance, there was one of our sites that it has like a lot of, for example, columnar corals, and there were also the presence of other coral that is very flat, that fungi corals. So then, for example, within a site, even the transects, they were different. So in one transect we found columnar, and then in the other we found, in the second transect that we deployed, we found this fungi coral. So that could be due to in this case, due to the biology of the corals, because this coral grows in the sand, and the other one prefers like a hard substrate. Yeah. And between the sites, for instance, I think that something that could be very interesting to explore is the topography, because it was different between the sites. That's something that we observed. And there were, for example, some sites that have like a pinnacle form. So they were a bit less deep, for example, than the other ones. Or there was the one that I mentioned, that it was a plateau form. And I do think that there are different factors that contribute to the corals. So that could be a lot of things like currents, like that, and a lot of factors that we didn't study.
I think you just mentioned one very important thing, the currents, because very complex shapes cannot actually be at places where you have massive, strong currents. They will break, and that's a physical limit that actually is described. I think there are some papers that relate current strength, current velocity with the complexity of the corals. So, do you have any measurement of currents, or could you extract that somehow? Do you know? Some models?
No, unfortunately no. And especially for this particular region that is very, is exactly between the two islands. I will go quick to the map. Okay. So particularly here, our sites were here. So it's very, small area, because we had like big currents coming from the south and then going in this direction in the Mozambique Channel. So that's the main currents in this region that indeed connect the coral communities here with the Seychelles region and Tanzania, but that's the like the big currents and main currents that contribute to this, and we don't know about this, the local ones that can link that.
We have to stop here. I had some questions and comments, but if you want, we can come and see me later because now we must stop. Thank you very much. Peter. Nancy, again, Nancy, Julie.
All right, hello everyone, good afternoon. Thank you for showing up today. My name is Ryan and I'm going to be discussing you the analysis of basking shark courtship behavior in Irish waters. Just as a quick disclaimer, the entirety of this study has not yet been completed, but we'll discuss that a little bit later in the presentation. So let's get a quick rundown of our focal species. So the basking shark, as you might know, is the second largest species of fish in the world, just behind the whale shark. And it is the largest species of elasmobranch within the North Atlantic waters. They do have a fairly wide distribution in temperate regions, both in the north and south latitudes. And they are known to have annual migrations in these North Atlantic waters during the summer months, ranging from the early April down to the late September periods. In particular, they are seen quite frequently along the Irish Atlantic margin, feeding towards the surface along the coast, and this is now being recognized as a designated hotspot for these animals. Despite their wide distribution and frequent observations, they are still considered an endangered species on the IUCN Red List, and this is attributed to their historically exploited past as well as their continued population decline. So like most elasmobranchs, their life histories and reproductive processes are poorly understood, given their elusive nature and the difficulties of doing in situ observations. However, we have started to notice some large annual social aggregations in recent years that are no longer related to feeding, which is quite unique for these animals. As you can see, not here. There we go. As you can see here, oh goodness. Okay. These large social aggregations are unique to these species, and they have been deemed a tourist formation by Dr. David Sims in recent years. Essentially what it is a large social gathering. We think it's directly related to courtship practices, where sharks can come in numbers of anywhere from 5 up to 40 individuals, which have been spotted, and they sort of form these tight-knit circling formations where they participate in close following behaviors and synchronized swimming events. The goal of this study was to expand upon what David Sims had found recently in this past summer, and to use ethology sort of as a tool of conservation in order to try and describe the different behaviors that were going on and provide a little more support towards the protection which has just been implemented for these species in Irish waters. This is the paper I'm referring to here if you care to look at it afterwards. The main objectives of this study were to understand the differences in the compositions of behaviors based on the group sizes of these sharks. And later on, we're going to try and find out the composition of the body size in terms of maturity, as well as the ratios of male to females within these courtship formations. So a quick overview of the study site. The majority of observations that were made were from County Clare in Ireland, which is just south of Galway, and they were all relatively close to shore. They were conducted during the later summer months of August to September. This tends to be when the sharks slow down on their feeding behaviors and they tend to aggregate in these large circular formations. The observations were made in 3 separate years in 2016, 2020, and 2021. However, for the focus of this study, we're going to focus on 3 tourist groups from 2021 alone. The 3 groups are as follows. Tourist 1 contained around 9 to 7 individuals. 2 of which departed from the video pretty early on, so they were vetoed from any of the data analysis later on. Taurus 2 was our smallest formation with around 6 individuals in total. And then our third Taurus group contained the largest number of individuals with 14 sharks. So the data collection and extraction period took place before I got there in those previous years, as mentioned before, by my supervisor, as well as the Irish basking shark group, who used a couple small inflatable hubs and the combination of drones and GoPro videos in order to gather some simultaneous recordings of these sharks, both from above and below. This allowed to identify any sex as well as any group size estimates that we may have seen sharks at deeper layers that we couldn't see from the drones above. While they were out there, they also took some field notes, including some tagging and some slime samples for later analysis. So the majority of the work that I did was sorting through these hundreds of videos and hours of footage and trying to select the best ones that I could use for some data analysis. And this was simply done using some Wondershare Filmora software in order to stitch together those videos and try and get the contrast and white balances better to see the sharks, as there's lots of glare coming off of that water. The first thing I did was take several still frames from each of those videos, and those were used later on to try and get some length estimates of those sharks whenever one of the small vessels was also in frame, which was used as a scale using some ImageStage software. However, this was only be able to achieve for 2 out of those 3 groups. The bulk of the work was developing the ethogram using Boris software. This is a behavioral event logging software that is free for online users. The first thing to do was to identify all the subjects that I would be looking at within each of the groups. And next was to create some formal definitions for the behaviors and attach any modifiers that might be linked to those behaviors. Afterwards, all of those ethograms, as well as a time budget series, was exported. And we used RStudio just to do a quick, simple data analysis. Here we looked at the frequency of events, which was simply calculated as the number of events per minute by subject, as well as the duration of events, and those were compared based on the different group sizes that we observed. So the ethogram development, as like I said before, was to identify the subjects. This was done by using the drone videos and looking for any permanent markings, scars, or even the tags that were on the sharks, just to try and give each individual a unique identification code. And again, the modifiers that were used here were the other subjects within each of the groups, as well as the direction of travel for each of the sharks. The events can be defined as either a point or state event. Point events are those that occurred at a single moment in time, where state events contained any behaviors that have a duration. The behaviors were subdivided into either interaction events or dynamic events, depending on what they were used for. Interaction events were logged as a binary event, and such that when Shark A interacted with Shark B was recorded as interacting with Shark A at the same time. So some of these events involved crossing under or above if a shark was to pass over one another. One of the big ones we'll be focusing on is following or nosing behavior, which is quite common amongst other elasmobranch species. And essentially, this is indicative of a male following a female closely behind her tail. And this was recorded as a state event to try and get the duration of that behavior. The final interaction event was any misdirections or any encounters, and such that one shark sort of skewed the other shark off of its original swim path. Dynamic events are those that were individual to that shark and didn't affect the other ones around it. So one of them we looked at was the increase in speed from its original cruising velocity. The next one was any changes in direction from the majority of the group formation of the torus, either clockwise or counterclockwise. Anytime the shark broke the surface with its dorsal fin, or anytime it dove to a deeper layer within the torus. Finally recorded any point that the shark exited the torus or entered or re-entered from an outside perspective. And as well as we recorded the number of laps, and such that when a shark completed one full rotation, it was logged down. Finally, the last state event was anytime the shark was recorded as being out of frame, as that had to be subtracted from the time budget analysis, as behaviors could not be recorded during that period. So some exploratory statistics. As we can see here, the 3 groups varied quite a little bit in terms of their frequency of events, with similar trends broken up into the interaction, dynamic, and lap frequencies, and such that Taurus 3 had the greatest number of frequency events, followed by Taurus 2 and then Taurus 1. When we look at the following durations, we can see that the occurrences of following were greatest in Taurus 3. However, when we break it down into our time budget analysis, we can see a little bit of discrepancy in such that the average mean duration of following from Taurus 2 was greater than those of Taurus 3. So what does all of this tell us? We did some non-parametric testing, which revealed significant differences between those groups. Taurus 1 and Taurus 2 had less significant differences, which was expected based on their similar group sizes. When we looked at the differences of frequencies of interaction events, they were different between T1 and T3, as well as T2 and T3. There was quite significant differences in the dynamic events between T1 and T3 only. And finally, there were differences in lat frequencies between T1 and T2 and T1 and T3. When we looked at the occurrences of following behaviors, T2 took a longer duration, T2 took a longer duration, but T3 had the most occurrences of those events. So, some less significant differences between T1 and T2 was attributed to their similar group sizes, and the differences between those and T3 was attributed to the fact that T3 had a larger number of individuals overall. This can tell us a few different things, especially in terms of interaction and dynamic events, and both provided insight into group dynamics based on the size and how they would work when there were other subjects around. The occurrences of following versus duration also gave us a little bit of insight on what sharks might prefer, depending on the number that are within a group, pertaining to the quality or quantity of those interactions and which ones might be valued more. So, some quick explanations, which have not yet been verified. We suspect that the largest Taurus groups have a higher frequency, which is potentially due to the social interaction and competition from the other individuals, and we're using these as essential a speed dating event in which each shark is trying to make sure it meets all the other ones within its circle. This can tell us a little about the group dynamics and organization, which may also influence the event frequencies, which is why we want to look at body length and sex ratios as well. The longer durations of nose and tail following behavior may indicate higher social statuses, which is why we want to look at some dominance hierarchies, as well as some reoccurrent following by individuals versus the quality may prove different benefits within those groups, and such that larger groups have more frequencies and that the sharks want to meet each potential partner, whereas in a smaller group, they might have a longer duration of that following period in order to try and coerce that female into copulation. So some continuations, limitations, and recommendations. As stated before, we briefly looked into body size, and there did appear to be a negative linear trend, and such that smaller individuals had less frequencies of events than larger individuals, indicating that there might be a dominance at play. For only T3, we were able to identify 10 out of the 14 individuals as being male or female using their claspers from the underwater videos, which we have not quite fully explored in depth yet. Using the modifiers that I stated before, we're trying to see if we can build some networks or social hierarchies within each of those groups and determine if there's any sort of alpha males or dominant individuals within those groups. And then finally, we want to look into the change of speed to see if the change in velocity is going to instigate changes of others, individuals around in that torus. So in summary, we have discovered some unique courtship behaviors, and we would like to investigate this further. This is going to be useful to help fill some knowledge gaps in shark courtship behavior and mate selection processes, which may be able to be used for other shark species. Finally, we hope that this provides some critical evidence and support as to protecting the Irish Atlantic margin as a hotspot for these species, and further create some more legislations. A quick thank you to my supervisor, Simon Barrow, as well as David Sims, Nick Pfeiffer, and the Irish Baskin Shark Group for helping out with the data collection and providing these valuable videos. Here are some references should you want to learn more, and I'll invite you for questions.
Thank you, Ryan, for your presentation. Just to say that you used 12 minutes and 30 seconds, so you had actually 2 minutes. You were a bit fast. You could have taken your time. That's fine. Just a general comment. I'm taking the time now because you left off the time, and a lot of you make that little mistake. Our studio is just an interface, right? Mm-hmm. You're doing your analysis in R. Yeah. Doing your analysis, saying you're doing the analysis in our studio is like saying I'm speaking in Olivier. No, I'm speaking in English. True. Olivier is the interface for English. Right. The language is English. The language is R. studio is
Is
the
the
interface.
software, okay.
Okay, so but all of you usually say
For
that.
sure.
Like 95% of you say that before we correct you. Karim, would you like to start the discussion? Thank you.
Okay. So you use drones to
Yeah.
get these pictures, and I noticed in the one of the pictures where you had the three Taurus groups, there was a boat very close to
Yeah.
them. I mean, doesn't that interfere with the behavior of the,
Yeah.
or does that boat belong to, or is it?
It does. So it was one of the boats that they used during the surveying. What I tried to do was try to select the portions of those videos where the boats were more at a distance. The boats probably were in frame at that point because they tried to do some tagging and some slime sampling at the same time. And the other sort of discrepancy there is that at some points there were snorkelers in the water, which were trying to take the underwater GoPro videos. So there's a little bit bias there of whether or not they may have interacted or interfered with, you know, the natural behaviors and movements and maybe scared some sharks away. So that's why I went through all the videos and tried to select the best ones where interference was mitigated, and we tried to, you know, ensure that.
Okay, so the hypothesis here, or one hypothesis here, that this is courtship behavior.
Yep.
Is there any evidence that this speed dating leads to any pairing and wander off or anything happening?
Yeah, so it's quite unique to these guys in terms of this like tight-knit circular formation. But because they come to Ireland every year, and in the beginning of the summer months, we see them open mouth feeding at the surface. Sometimes they're alone, sometimes they're in these aggregations, but they're swimming more parallel to each other, and they're not creating these, you know, these donuts in the water. We do suspect that it would lead to courtship because there's not much other behaviors that we think a group of sharks would be doing. But that being said, some of the behaviors that we do see, especially nosing or following, is quite indicative of other shark species that do that and has definitely led to copulation and mate selection. Normally it's a group of sharks, especially sand tigers and nurse sharks. There's normally a sole female that are being pursued by a bunch of males, but they do that following behavior where they try and get their nose as close to the tail. Maybe it's because they're releasing pheromones and they're trying to determine which female is, you know, most fit for copulation at that moment.
And this circling goes on for how long?
Yeah, so that's the tricky part, is that the drone battery can only last for 20 minutes. And then because they're doing these surveys and they are looking for these circular formations, we don't quite know when it has started. And then based on that observation day, we don't know when it would have ended. So it could go on for hours and hours technically, from what we know. What we were hoping is that maybe we would start to see two sharks pair off and break off, or maybe there's sort of a dominant individual that kind of instigates behaviors, and the formation of the torus, we haven't fully, you know, got that fully on film in the full entirety completion of it. Most of the videos just started kind of halfway through, or at least what I would suspect through the actual donut.
You're not doing any sort of like using chat tags or anything like that to see who's interacting with who?
Yeah, it would be a good idea. Most of the tags that they were using were just plastic Casey tags, just for a unique identification, maybe for mark recapture purposes to see if they're going to Canada or to see if they're coming back every year. But it would be good to use those tags if I could get close enough to see the numbers and see if the same sharks are coming in the same formations. With this study, we had to assume that each of those sharks was a unique individual, just because we couldn't fully see the number. But
I was meaning these tags, that electronic tags that,
yeah, that
they
ping
come,
off each other.
They can tell you which one has been in contact.
Exactly. Yeah, we don't have those. So the only other way we would do that is to get maybe unique identifications or pigmentations or scars and see if those sharks are constantly coming together based on all the videos. But it's kind of hard to tell sharks apart from video to video.
Nico?
Yeah. First of all, congratulations for the presentation. I think it was quite clear. Also the way you talk, it was very nice. A little bit too fast, probably.
It was.
We was able to understand everything, so very well done. And about the science itself, I think this is a very interesting topic, quite new actually. And I was wondering if there would be a way to identify the dominant guy.
Yeah.
Just by shifts, little shifts in the trajectory.
Yeah. Yes. So when I use the modifier, so every time there was an interaction event, I would be prompted to say which shark was interacting with which shark. So later on, I would like to develop a network and see if one shark is interacting more frequently with all of the other sharks, or if they're sort of, you know.
I was thinking in tracking like little changes of speed and direction to see who starts first.
Yep.
And if the other
They
few seconds
follow.
later repeat the same.
Yeah.
And that will rank your animals in a kind of
who's initiating the movements.
And who follows.
Yeah, exactly. When you're looking at them very slow, like how you saw in that video, they are quite slow moving. But when you play the videos on 2x speed, you kind of do see patterns and such that if one shark breaks off, they kind of all start to follow that shark. So I took notes of that. There was no real way for me to log that behavior unless I put the shark exited the torus. So if I were to lay out the ethogram with all of those behaviors in order based on subject, there could be a way definitely to see who initiated that and then who followed pursuit. Yeah.
But what were your initial expectations? And what are your thoughts now? Do you think it's a kind of way to address the different hierarchical levels of these sharks? Do you think it's a shark Tinder or something like that?
Yeah.
Or
yeah, it's like
do you think that both are possible, or what do you?
Yeah, so what the biggest one we would try and do is trying to identify as many males and females as possible.
Yeah.
The challenges I had is that the GoPro videos underneath don't really let me know what the drone is seeing above, because the drone, I'm looking for scars or tags to try and point out those individuals from above. And then below I see their bellies, so I don't know really if that's the same individual. We only managed to do it for a couple. But yeah, definitely for sure with those hierarchies as well, we could sort of begin to map that if we get a little bit more data and run those through.
How often do you observe these?
The torus. So the sharks come in like beginning of April around the Irish waters, but they're feeding, and then towards the late August, early September is when they seem to really stop feeding and they start to focus on these circular formations, and they sort of gather together in bunches. There was a team in Canada that saw upwards of 1,000 sharks, so they can get quite big. The ones we had here averaged around 10 to 15 probably over those 3 years. But they can be as small as 5 sharks all following each other in perfect unison, or we had 40 sharks in one of them that I'm trying to slowly pick away at.
Thank you very much.
All right. Thank you, Nico. Yes?
Yes. Thank you, Ryan, for your presentation. It's quite interesting for me because they are doing that. But my question first is, are there genetic studies on the familial composition of this group? Is there something about this?
Genetics in total?
Yes, that could give us some insights about what is the familiar.
Yeah, little genetic information has been done. I think that's what the team was trying to do with the slime samples as well. I think we still need a little bit more. There was a genetic analysis done to see sort of their migratory paths, and all they really found was that there's a good group of sharks that go from Canada, and then they go sort of transatlantic to the Irish waters. So in terms of genetics, that's about as far as they've gotten in terms of that, and it's really been focused on distribution patterns.
Because I was wondering if they are coming from different places, and
Yeah,
then this is the,
right?
Because maybe this is a previous meeting before.
Yeah, exactly.
You know, this
Exactly.
like know each other.
Yeah.
The group, but they are different. They are coming from different places, and they are this kind of.
For sure. Yeah, it would definitely be interesting.
How was the sex distribution in the groups?
In the ones that we did get?
Maybe I didn't get it, but the balance between sexes is the same in small groups that in larger groups?
Yeah, so we don't quite have that information yet because we only managed to identify the sex of 10 individuals out of the 14 in that third torus. So we only have 10 out of 14. I think there's a higher population of males within the groups. But yeah, we would like to see the ratio of males versus females depending on how many are in a group, if there's more males when there's only one female, or if more females will come if there's more males, or.
I think that it was very interesting to know more about the behavior of them. Because at the beginning, when you were starting, I was thinking, but it's obvious, no? I mean,
Yeah.
I mean, if the group is different in size, I assume, thinking, I mean, guess that I am just your brother. I mean, your brother.
Yeah.
Know nothing about this. I mean, I'm expecting that if the group is different, interaction and the behavior will be different.
Yeah.
If you have only six people, it's different than when you are interacting.
Of course. Yeah.
So what is the meaning behind that? I mean,
yeah, so what we want to try and do is see if there's any sort of mate selection processes. So trying to get a little bit more metrics on the sharks themselves, so their body size, their maturity levels, or their genders, and just seeing if the group size sort of affects the differences in behaviors. The big one's going to be looking at males. If there's more males within a group, does one male tend to dominate over those females? And if it did, you would start to see more interactions from that male. And then as a result, it would have more dynamic movements within the torus in order to try and jockey positions around the females and corner off the other males.
okay, thank you so much.
No problem.
All right, thank you, yes. I think we're gonna stop here. Thank you very much, Ryan. Just one thing it wasn't said, I must say it. In terms of it's a comment we often make, the size of the fonts.
Yeah.
You were a very good example of what not to do.
Okay.
Unfortunately, regardless of the quality of the work and the presentation, even the top frieze, like
Yeah.
just the name of the section, was, I'm sure from behind,
Difficult.
it's barely readable, and in the figures it was just impossible.
Okay.
Really watch out for that.
Yeah.
Again, put forward what we must see and hide it. If you don't need to see, just hide it.
Okay.
All right. But other than that, congratulations.
Gotcha. Thank you.
So yeah, we have. whenever you're ready.
Okay. So hello everyone, I am Léa Sauvanet, and today I'm going to present you my master thesis, which was about the variability in the lipid composition of Cerastoderma edule and associated species along an intertidal gradient in the Bay of Saint-Brieuc in the English Channel. I did this work under the supervision of Gauthier Charles at l'EMAR at Clouzavez in France. So let me introduce you to a harsh habitat, the intertidal bay. So it is an ecosystem under a lot of pressure. The first one is the wave exposure. In fact, in my example, we are in Brittany, so it's a semi-diurnal megatidal regime. So the sea level rise from this area to the upper part of the shore 2 times per day. So it induces a tidal current, and also there is also a lot of wind driving soil. In addition to that, there is a difference in the sediment composition along this gradient. In fact, the more sheltered part here, the salt marshes, the sediment is composed by a fine particle, like mud, while in the more exposed one here, it's a sandy layer. And in addition, there is, as I told you, various immersion time. So all of this will allow an intertidal environmental gradient with a lot of variation in temperature, but also in humidity, in oxygen. And all of this will allow biotic stress, because in this area we will have competition for space and also a lot of predation. So to understand this ecosystem, I worked on lipid, I work on fat. Why? It's a good question. So you need to know that there is 2 main categories of fatty acids. The first one is a membrane lipid, which is composed by polar lipid classes. They are the main component of the cell membrane. They are composed by several polar lipid fatty acids. These fatty acids are under metabolic control, and they can reflect the vital physiological processes in an organism. It also highlights the seasonal variability of the diet. On another hand, we have the reserve lipid, which are made of neutral lipid classes. They are used to store their energy into the body, and they are composed by neutral lipid fatty acids. These fatty acids are not modified after the assimilation by the consumer, and some of them, the long-chain fatty acids, can be used as a biomarker. In fact, the carbonate chain are too long to be biosynthesized de novo by the consumer, so it remains intact along all the trophic web, and it can be a great tool to understand what the consumer feeds on. But let's go back in Brittany. So this is Saint-Brieuc. This is a contrasted bay. In fact, as I told you, there is different habitat, the sheltered one in the salt marshes, but also an exposed one. There is different food supplies also, like phytoplanktonic algae, but also benthic diatom, detritus in the sediment, and as well the salt marsh detritus. There is also, as I already told you, a lot of biotic and abiotic stressors. So all of this will transform the benthic assemblage in this area. Saint-Brieuc also has a national and international interest. In fact, it hosts 1% of the international wintering shorebird. And me, I worked on cockle, Cerastoderma edule, because this species is widely distributed along the bay, and also this is a key species supporting the shorebird population. So the aim of my study was to understand the trophic ecology of Cerastoderma edule, the common cockle, in Saint-Brieuc by understanding first the main potential food source, and also second, to understand the seasonal physiological regulation of this species and associated invertebrates facing this contrasted environment. So this is my area of interest. We are in France here, in north of Brittany, in front of the English Channel. Here on the map, this is the left part of the Saint-Brieuc Bay. So we did a transect from the sheltered part here to the exposed part here. We did 4 stations with triplicate. We did 2 sampling, one in January to understand the winter lipid composition, and one in April to understand the spring one. We sampled 3 species. Here, Cerastoderma edule, but also Scrobicularia plana, another bivalve, and Perengauve, a gasteropod. Okay, so to understand this sample, I first extract the lipid using chloroform methanol solution, and after I did an HPTLC. So I used this extract, And I did several depositions on the silica plate. And after that, I scanned it into the scanner. And at the end, I had this kind of chromatography, which discriminates each lipid classes depending on their wavelengths. So I had the lipid classes. To go further in the analysis, I did a gas chromatography. So I used the same lipid extract, but after I did a derivation, which is the composition of a saponification and a transmethylation. So at the end, I had a solution called FAME, which is fatty acid methyl ester. It's more volatile, and it can pass through the GC column. So after this analysis, I had this kind of chromatography, which is the different fatty acids that are discriminated depending on their retention time in the column. And after that, I could have know the concentration using an intern standard. Here's the C23. Okay, so this is my first result. This is a spatiotemporal variation in the lipid classes of Ceratoderma idulae. So in the y-axis, you can see this is the relative lipid class composition. And here, this is the station from the first to the last in winter and in spring. So in overall, we can see that the lipid composition are more or less constant over the time and the space. And also, the main component is always the orange layer, the P plus Cl. Okay, but here we can distinguish three anomalies. The first one is that in the first station is showing the highest lipid content here in comparison with the other station. The second one is that in spring, we have a decrease in lipid classes along the gradient. And the last anomaly that you can see, that in winter, it's characterized by the presence of PE plus CAP, and there is no triglyceride, which is the main lipid class of reserve in bivalve, while in spring, there is a lot of triglycerides, so there is a lot of reserve. Okay, so to understand the fact that the first station shows the highest lipid content, you need to know that this station is at the vicinity of salt marshes, and salt marshes are systems that produce a lot of organic matter. So it is a significant food source available quickly for the surrounding bacteric assemblage. And the fact that there is a decrease in summer in lipid content could state because of the outwelling hypothesis, here set up by Odum in 1968. This hypothesis states that the salt marsh are systems that produce a lot of organic matter, a lot of material, that it can't store it itself or even decompose it. So it creates a flux from this salt marsh to the coastal ocean with a decrease in lipid content. So remember what we say about the triglyceride. Here, this is another graph showing in winter here and in spring here the concentration of membrane lipid in orange and in yellow the reserve lipid. So we see the same thing, that in winter there is no triglyceride, no reserve. It could be due because there is a nutritional deficiency at this time. In fact, we are in a cold season, so there is less primary producer like phytoplankton, but also salt marsh detritus. So what the cockles feed, they consume it directly without storing it. So all of the reserves are exhausted. And the fact that we have a lot of PE plus CAP, the green layer here, could be due because they need to increase the hemocyte into the system. And hemocyte allows a lipid raft into the cell, which is used to strengthen the immune system of the cockle. While in spring, we have a lot of reserve lipid here, the yellow. It's linked to the reproduction cycle. In fact, the gonads are doing a lot of reserve to have a lot of energy to after produce the gonad between April to May, because we did the sampling in the beginning of April, and after doing the spawning in summer. Okay, this is another result. This is a fatty acid composition of the common cockle along the intertidal gradient. So this is a principal component analysis. The first dimension is showing 20% of the variability, while the second one is showing 12%. So on this PCA, there is different ellipses which are showing the different station. In blue, this is the first station, yellow the second, red the third, and green the fourth. So here, this is a different fatty acid. So we can see, first of all, that there is a spatial gradient from the first station here to the fourth here. But we can see also that the first station, first and second, are correlated with trophic biomarker. So here I choose several, like the one for diatom, one for bacteria, and another one for higher plant. So it means that the first station fatty acid composition reflects the dietary input, while the last station, the third and the fourth, are more correlated with fatty acid with a metabolic origin, like here the DMA, which is used to increase the concentration of plasmogen into the organism. And plasmogen are used to maintain the cell membrane integrity against biotic stress, like viruses, bacteria, and also cancer. And also abiotic stress, like an increase in the temperature, a decrease in oxygen, like hypoxia, and also a salinity variability. So this is my last result. In blue, you can see this is Cerastoderm idule here. Here in purple, this is Peringium V, and in orange is Curbicularia plana. So this graph is showing the concentration of this specific fatty acid, which is marked for terrestrial plant, and this is the second main component of cypress land at Saint-Brieuc. So you can see easily here that from the first station to the last station, there is a decrease of in the concentration of this specific fatty acid, and it is the same for the three species, which means that the population living in the sheltered area in the first station are using cypress lane and are using salt marshes as a food source, while in the second row, this is another fatty acid here. This is this one, which is marking for dinoflagellate and broadleaf for phytoplankton. And you can see the opposite here. It's increasing along this gradient. So it means that the population living in the last station are using a phytoplanktonic food source. Okay, so what did we learn during this study? We learned more about the physiology of the common cockle. We know that they need to strengthen the immune system in winter, and also they need to store a lot of energy, a lot of triglyceride to do the gametogenesis in spring. We also know more about the cypress lane. In fact, the cypress lane are one of the main food sources for the population living in sheltered area of the cockle, and cockle are one of the main food sources for shorebird at Saint-Brieuc. So it means that the salt marshes contribute to the functionality of the bay for the bird and also for the surrounding biotic assemblage as a habitat, but we knew that already, but also as a food source. The last result is showing that the population living in this area, in the salt marshes, are using cypress lane, the vascular plant, as a food source, while in the exposed area on the sand, the population are using phytoplanktonic food source. If you want to go further in the analysis, I think it could be interesting to do a CUFSA analysis, which is used to understand the lipid composition of predator. And I think it could be interesting to do that at Saint-Brieuc on the shorebird, because if we know the impact of the cockle on as a food source for the shorebird, I think it could be way interesting. And also I think it could be interesting to extend this kind of analysis throughout the year, because in fact in Côte d'Armor there is a lot of, where Saint-Brieuc is, there is a lot of agricultural activities, and because of the runoff, a lot of nutrient is going to the coast, and I think it would be interesting, with the point of view of the lipid, to understand how these benthic assemblage are coping with the new anthropogenic stress, the green algae bloom today. Okay, thank you.
Thank you, Leah. You were just under a minute of left time, so pretty good timing. Thank you for the presentation. It was very clear. And Gauthier actually was here, and I can say that he was connected, and he would like to congratulate you for the nice presentation. He's proud of you. So let's start with that. Okay, and then I will let. We didn't start. Nico, you never started the discussion in this session. So Nico will start the discussion now. Yeah.
Yeah, I want to join this congratulations because you really deserve it. You made a very nice presentation. I really like the way you make yourself some questions. And after a few seconds later, you reply them like, I think, I like it actually. It's a good way of letting the public know what are you actually talking about. So it's good. And also the results are explained in a very clear way. The figures are nice, especially the maps. Although you are having the same problem as many others with the size of the legends.
Yeah, I saw it here. I was like, yeah, it's very small.
And sometimes it's not important, and sometimes it is very important, like here. Because here you mention the names of the species.
Yeah.
But you mentioned that, but they are not there. They cannot see.
Yeah, you cannot see it here. And I put some picture.
Yeah, but anyway.
Yeah, I know. I saw it. It's very small here.
Yeah.
The PC was worse, if I may say.
Yeah, the PC is worse.
And there is a previous graph. If you go back, I think there is another bar graph. No.
The first.
Yeah, this one to the right. The one to the right. What are those treatments? What is orange and what is?
Yeah, it's here, but it's very small. It's membrane lipid classes here. It's all the lipid of this category. And in yellow, it's a reserve, but I agree. Can't see.
Yeah, it's a pity.
Yeah.
I mean, the presentation is good, but this details.
Yeah. I saw it when I did the presentation. I was like, yeah.
And about the scientific results that you're presenting here, I lack a little bit, miss a little bit of more information about the biology of the target species of the cockle. What is the habitat across the intertidal? Does it get to the subtidal? At which depth is the cockle living within the sediment? That's an important parameter.
Yeah, in few centimeters, but also it changes depending on the tide, where they are in the sediment.
Yeah.
They
can go up and down.
Yeah, and they are filtering the water, so they go more.
And the other species that
Sorry?
you are comparing with the other species?
The other species, so the Peringeolbe, the gastropod, is living under the sediment. Is living on the sediment, above the sediment.
Okay.
And Scobiculea plana, this is the same, more or less same as the cockle. They are living in the sediment in few centimeters.
Yeah, those details can be useful to explain the patterns that you can find. Yeah, I don't have more questions. It was a good presentation. Thank you very much.
Thank you, Nico. Yes? Follow?
From that layer, we are happy for you stay here. Are you interested in Yeah. Okay, just very general questions. Do you have you any example about how the lipid composition of this species has been used for a better management, a better conservation policies in some areas? I missed in your presentation like previous background about how to use lipid composition for doing something that will be useful, for the conservation and management.
Yeah, in fact, at some years there is a lack of. It's the first time we are conducting the lipid analysis. Isotope analysis have been already conducted there. Someone did a PhD on this, and yeah, this is like a first step of lipid analysis at Saint-Brieuc. And so yeah, I don't have a background study before in this area. And in fact, there is a natural reserve in this area, so it's interesting for the management of the area to understand the lipid composition. But it's like the first step here.
And why did you choose these three species? I mean, because it's clear that you were focusing on this one, but why the other two? I mean, what was the reason behind that?
Okay, so when we went sampling, we didn't know which species we would choose. So we took what we found, and the cockle was the one which was so widely distributed within the bay, and we had a lot of samples, so we took the cockle. And for the two other, it was the same. It was only two other species that we found in several stations, because we wanted to see the variability along the gradient, so we wanted to have sample from all the stations. So yeah, Peringeolbe, we found found it in 3 stations, and Scubicularia plana in only 2, but it was only 3 species that we found along the gradient.
So it was common and you can compare.
Yeah, we can compare.
Species, the mechanisms. So the sampling was from January to.
No, sorry, it was 2 sampling different.
Okay.
One in January and one in April. It was just 2 snapshots.
Okay. Okay, so it would be interesting, as you said, it would be interesting to complete the annual.
Yeah.
Okay, no more questions from my part.
Right, thank you. David?
Okay, thank you. Yes, I'm from the Algarve, in the south of Portugal. We have a lagoon where the cockle is extremely abundant, but I didn't know it was an important food source for wading birds.
Yeah.
And this, I assume, like in Rio Formosa Lagoon, this is an important place for migratory birds where
Yeah.
they restore their energy. I was just wondering, do the migratory periods coincide with the period where they have the highest lipid fatty acid
No,
contents and all that?
This is the opposite, in fact, because the shorebirds are going at Saint-Brieuc in winter because it is in the middle of the Atlantic Channel, I think, in the migratory way. And this is when the cockle they don't have any storing lipid because it's cold, so I mean they can't storing a lot of things. But this is because I think because there is a lot in this bay, this is one of their main food source at Saint-Brieuc because there are a lot.
Okay. One thing I didn't quite understand, but maybe you could explain it to me a little bit better, is about the need to strengthen the immune system in winter. So
how can I know that?
No, can you explain that? What happens?
Yeah, it's a normal question. So the first thing, it's because in winter there is this specific lipid classes here, and it has been discovered at Brest also by this person that it increases the percentage of hemocytes in the cell, and hemocytes they are used to create lipid raft, and raft they are used to how can I say that? When there is a lot of viruses and also bacteria in the ecosystem, the cockles need to have a lot to increase these lipid classes to fight against these specific stresses. So by this way, I can say that they need to strengthen the immune system because they show a lot of these specific lipid classes.
So could that be related, for example, to a pollution gradient along the?
I can't say that. I don't have the data to say that.
And you said that stabilizers have been used already to look at the trophic ecology.
Yeah.
And so what those results, do they tie in well with your results and what you found? The cockles feeding on the, what do you call it, vegetation higher up.
But the isotope analysis was more to understand the whole trophic web at Saint-Brieuc, while me, I focus only on few species and mostly on cockles. And this time during this study, he didn't know, he couldn't have shown this particularity that cockles feed on the detritus of cypress land. He didn't know before. And thanks to this analysis, we can say yes. But it wasn't on the paper.
Yes, I did. I thought they were more filter feeding plankton rather than the dreyfus.
Yeah.
No more questions? It's the first for me today. We have time for questions from the audience. Okay, go ahead.
I forgot. What shorebirds are we talking about? Oyster catchers, wingows?
Yeah, oyster catcher.
Oyster catcher.
But I'm not a specialist in birds, so I can't really tell you.
Okay.
You ask Jacques to add one.
Yeah, you would know.
Any questions from the audience? No? Okay, from online, we also have Aliyah cheering for you, and Léa Genet from
Yeah.
congratulating you.
Thank
So
you,
I think
Léa.
with this I think everybody is tired, the presenter, the students, the juries, the tech support. So I think we're gonna stop for today. First of all, Léa, congratulations. Congratulations to everybody that presented today. Be nice to your fellows that present tomorrow, and the residents, and all the best of luck for those that present tomorrow.